Hello, and welcome back to the Biology 219 video playlist. Recently, we have been talking about the importance of DNA and how it is stored, maintained, and replicated. Now, we'll talk about what it is used for. Remember that DNA is information, like a recipe. A recipe is only useful if it can be used to make a product. But DNA is also a coded message, and cells can only use DNA to make products if they can understand the code. The DNA code has several features. Parts of the code specify products, but parts of the code specify which cells should make the product, when the product should be made, and how much product should be made. For example, stomach acid is made of mostly hydrochloric acid with a pH of about 2.0. There are proteins that make and secrete stomach acid from stomach cells into the hollow part of the stomach. It's important to make these proteins in very high quantities in the stomach, especially after a meal. But it's also really important not to make these proteins in other cells, like muscles or the cells of the eye, where they could do a lot of damage. Part of reading the code to make products is also reading the regulatory sections of DNA. A second feature of the DNA code is that the order of nucleotides in DNA specifies the order of nucleotides in RNA, which specifies the order of amino acids in the protein. The code is triplet, which means that 3 nucleotides are used to code each amino acid. These 3 nucleotide sets are called codons. The code is also degenerate, which means that sometimes more than one codon can specify the same amino acid. For example, there are 6 different codons that all code for the amino acid serine. Finally, the code is non-overlapping. That means that an individual nucleotide can only be part of one codon. Let's see how this plays out in the cell. Let's say we wanted to make the pepsin protein used in the stomach to break down other proteins from food. The cell starts by finding the region of DNA that codes for pepsin. We'll talk a lot about this process in the coming weeks. Once the cell finds the right region, it has to determine which is the coding strand and which is the template strand. The enzyme for transcription reads the template strand to make a new RNA copy that is nearly identical to the coding strand. The enzyme that performs transcription is called RNA polymerase. It's similar to DNA polymerase from replication, in that it reads the template 3' to 5', so it can build a new strand 5' to 3'. Remember that RNA and DNA have a lot in common, but the 2' hydroxyl group that is missing in DNA is present in RNA. And RNA molecules contain uracil instead of thymine. Now the cell uses its code to read the mRNA and make protein. This process is called translation, and we'll get into the details of this in class soon, but for now, let's just look at how to read the codon table. First, the cell figures out where to start the protein. Proteins always start with methionine, so the code should read AUG. The ribosome will read the mRNA and place the appropriate amino acids according to the code. This code will start with AUG and methionine. Because the code is non-overlapping, the next codon is AAG, which codes for lysine. Then, the next code is UGG, which codes for tryptophan. And the ribosome will continue reading until it gets to a stop codon. In this way, the DNA code specifies the protein code. Some changes to the DNA code will have consequences for the protein code. For example, if the first codon is changed from AUG to AUC, isoleucine will be placed instead of methionine. This is known as a missense mutation. If the third codon is changed from UGG to UGA, a stop codon will be read instead of tryptophan. This is known as a nonsense mutation. If a nucleotide is inserted into the code, each codon frame will be off by 1. This is known as a frame shift and can also occur if a nucleotide is deleted. Finally, because the code is degenerate, some mutations have no consequence. Changing the 4th codon from CUG to CUC would not change the amino acid. Both of those codons code for leucine. In class, we will discuss the experiments that demonstrated these processes and get into the details of how they work to ensure that each protein is made at the right time, in the right place, in the right quantity to keep organisms functioning and healthy.